Hello, everyone, and thank you for joining this month's Bench to Bedside webinar series. I'm Nicole Wilson, Program Manager for the United Mitochondrial Disease Foundation. We're so glad you're here. Before we get started, I would like to remind everybody about MitoShare, our worldwide patient-populated registry. This is the ideal place to help capture the overall journey that our patients experience. So please let your patients know about this important registry today. Save the date for Mitochondrial Medicine 2026 in Orlando, Florida, June 17th to the 20th. You can learn more at umdfconference.org. We look forward to seeing you there. And now I'd like to welcome UMDF's very own Science and Alliance Officer, Dr. Philip Yeske, along with the moderators for today.
Well, hi, everybody. Let me begin by wishing everyone a very happy and healthy new year. Welcome to 2026. It's really exciting to be able to kick off the year with additional clinical trial updates in the mitochondrial disease space. As many or most of you should know, we did a first session back in December of 2025 that can now be found online, was recorded. And it's really exciting that there's so much clinical activity taking place, therapeutic development in mitochondrial disease, that we actually need two sessions to be able to cover the entire space. So today we're going to get updates from seven different organizations. I'm going to moderate and kind of walk you through the speakers here today. We won't be able to accept any questions as a part of this session. But obviously, if you're having any technical difficulties, don't hesitate to reach out over chat, and one of my colleagues will help you, correct any issues there. So why don't we get started? We're going to begin with a couple of regulatory updates, and the first is from Sharon Ham, and she is the Senior Vice President at Sale Therapeutics. And Sharon, I'll turn it over to you for your update.
Good afternoon, everyone. I'm Sharon Ham. Happy New Year as well as we kick off the year. I'm from Cell Therapeutics. And next slide. Today, we'll have an opportunity to give you a brief update as we look at the drug SL1009 for the treatment of PDCD, both from a regulatory and clinical perspective and continued access to treatment. For starters, PDCD, or pyruvate dehydrogenase complex deficiency, is a genetic defect that results in reduced PDC, or pyruvate dehydrogenase complex activity. As you can see from the graphics below, it's a refresh on your cell biology. What you see here are the differences in healthy human activity in the Krebs cycle as compared to what it would be like for a patient with PDCD as opposed to those patients then on treatment with SL1009. In the case of SL1009, this is a drug whose activity inhibits the PDK and results in increased PDC activity where there's residual PDC activity remaining. It acts in effect as if it was an enzyme replacement for that pathway, enabling an increase in the amount of energy available for metabolism. And excuse my residual Christmas cold. We had two clinical trials that were a part of the NDA submission. 01 was a double-blind placebo-controlled trial that had both double-blind and open-label phases with patients aged 6 months through 17 years of age. 28 of those patients still remain on the open-label study, and some of those patients have now been on therapy for a considerable amount of time, up to 4 years. The second study was a survival study that actually compared the outcomes for patients treated with SL1009 from the first study against a natural history cohort of patients. And the outcomes of that data set demonstrated that DCA, or SL1009, was indeed very well tolerated, even after chronic administration in close to 4 years of treatment. The primary efficacy endpoint that was identified for the study was a motor function scale using an observer-rated outcome. While that did not demonstrate statistical significance in the double-blind portion of the trial, a subset of those patients who had a greater impairment using that scale actually did demonstrate a significant improvement. In addition, we had significant improvements in plasma lactate concentrations, and we saw improved survival. And we'll cover some of that in a bit more detail in the slides that follow. As many people are aware, we filed the NDA with that data package, and then we received from FDA in August a complete response letter. The agency's primary focus for the complete response letter was, in fact, very focused on the clinical program of activity. In Study 01, they identified that the primary endpoint had not been met. As I mentioned, we identified as well. And that there was some discussion over higher baseline patients and the appropriateness of them relative to their use in the treatment effect. Secondary endpoints, lactate was met. However, there was some question in the agency's mind regarding how to identify that from a clinical benefit perspective. In the O2 study, we had an improvement here as we reported it, but the agency had questions about the external control, some of the diagnostic methods and advances in care since the time of the original development of the natural history cohort data set. And so these were some of the challenges we had, which led them to suggest that we needed to have an additional adequate and well-controlled study. As a result of that correspondence with the agency, we had some subsequent engagement with them, requested, and then undertook a Type A meeting in the run-up just to Christmas. That Type A meeting gave us an opportunity to share with them additional analyses to address some of the issues that they'd identified. And in particular, some of the points that were raised were specific to things like a functional benefit, where we provided additional analyses of the functional data, and were able to show some of the longer-term treatment effects on functional benefits, the survival benefits, additional analyses to address the questions that they raised, and to demonstrate the benefits of that continued based on our analyses. Mechanistic support in terms of the demonstration of how SL1009 acts mechanistically relative to the underlying causal issue for PBCD. And safety, because by now we have, as you might imagine, over 100 patient years of exposure and a well-demonstrated safety profile. We had the meeting with FDA, and as a result of that meeting, we were requested to undertake another Type C meeting to discuss in more detail the new survival analyses, give them an opportunity for a more detailed review, and then to also discuss the resubmission activity that would follow that without the need for an additional clinical trial. With respect to some of the additional analyses, what we demonstrated from a functional benefit perspective in double blind was that those patients who had motor scores higher than 6 had a significant improvement over the double blind treatment, as well as continuation over longer term exposure. The longer term treatment duration, as you run into open label, again, we were able to see clinically significant improvements in ClinRO gait performance and in Karnofsky-Lansky outcomes, which are physician measured endpoints that occur at study visits. And the most notable changes that we've seen in functional performance occurred as you kind of continue to look over time, which you see in this graphic display, where patients have been out on therapy, and over time you continue to see an improvement in their motor function. With respect to survival, what we saw was a reduction in death ranging from 97 to 99%. We undertook a range of additional sensitivity analyses in an effort to address the questions that the agency raised. And across those findings, we continue to see a strong survival benefit. There were additional analyses for lactate We demonstrated a significant reduction in plasma lactate in the double-blind portion of the trial, and in addition, continue to monitor over time. But further than that, we see amongst our emergency IND patients who are treated and have much higher baseline lactate levels, a rather significant reduction from much higher baseline plasma lactate levels that also is beneficial for patients. SAIL continues to have access available to SL1009. Our clinical trial patients continue in open label and are ongoing in terms of the open label portion of the trial. Further, we support emergency use IND treatments, and those activities continue. And expanded access is also now open for all patients of any age who have a confirmed diagnosis with PDCD. And we have sites that are actively enrolling patients. If there is an interest, you see here the contact information if there's an interest in getting to know more. As for what's next, obviously from sales perspective, we'll be now working in preparation for an upcoming Type C meeting and supporting resubmission activities. We'll be continuing to support access to SL1009. And obviously in the future, we look forward to engaging with FDA for what we hope will lead to their regulatory approval for this treatment in this new year. Thanks, everyone.
Thanks so much, Sharon. Very appreciative of SAIL continuing to try to bring this investigational therapy to market for our PDCD patients. And obviously, we'll be watching with great interest of the progress that you make here in the coming months. So thanks for that update. All right, we're going to turn to Giovanni Paciatori. Giovanni is the Senior Director of Medical Affairs for Chiesi Global Rare Diseases, and he's going to give us a brief update on the progress they're making with their investigational therapy. Giovanni.
Thank you for having me today. Sorry. I'm also fighting a bad cold.
It's that time of year, yes.
Yeah, sorry. It's like it's a winter, right? So my name is Giovanni Paciatori, and I'm the head of the North American Affairs for Chiesi. Today, I'm here to provide not really an overview of our clinical trials, but more a regulatory update and our milestone that we just achieved, especially while we are trying to getting the first approved therapy for Leber hereditary optic neuropathy in the US. LHON is a genetic disease affecting mitochondria, which present painless loss of vision acuity, also usually leading to permanent blindness within a few months. First off, though, I would like to introduce Chiesi Group and Chiesi Global Rare Disease to this audience today, because we actually have a long history. So Chiesi is a family-run business with a long history in discovering and developing medical solutions. So we actually, in 2025, we celebrate our 90th anniversary. So we've been there for a while. So, and it's not just about innovation with Chiesi. We are really committed to productivity and responsibility, which has earned us many certification and recognition, something that we are very proud of, and in our opinion, is very impressive. Chiesi is the largest pharmaceutical group in the world to receiving the B Corp certification. So what is a B Corp? B Corp, it's a certification that means that we're taking very seriously balancing the purpose and the profit. It's actually in our bylaw. It's regulated by Italian and US law, which every consideration, any decision that we made, take in consideration the impact on employees or customers, suppliers, community, yourself, and the environment. So some other companies that are in the B Corp, like the most famous probably are Patagonia or Ben & Jerry's. So, but just to see that that's for us is a very important part of our vision and our commitment to the community. So also the Chiesi Group, when it comes to strategic focus, is centered around three main areas: hair, care, and rare. So hair and care are the most likely the legacy area, and hair is about all respiratory health, while care covers spatial care and consumer health. And rare, the one that we worked, is dedicated to treatment to rare, ultra rare disease. But we also are really investing in research and development to make a difference in these fields. Up to 40% of our rare disease revenue are reinvested in R&D every year. And for rare disease, which is like where I work and is a topic here today, we founded actually was founded the Chiesi Global Rare Disease in 2019 with a few employees. And right now we have more than 700. So we grow pretty quickly in the last 6 years. And we are a direct presence in more than 30 countries, and we reach more countries through the Chiesi Group, the larger Chiesi Group. And we start in the rare disease because we understand there was a really need for treatment in this space. 95% of rare disease, as you guys well know, don't have a treatment options. Half of them don't have a patient support groups. 70% have a pediatric onset, and overall they affect more than 300 millions. So rare, it's rare for each single individual probably, but overall 300 millions doesn't look so rare to me. And only 30 million, actually, approximately 30 millions are in US only. But also in our rare disease unit, we are focused in different areas. Inborn error metabolism, rare hematology and immunology, rare ophthalmology, which I'm going to talk about later, which LHON is part of, endometabolic condition, and rare dermatology. So we have got an innovative pipeline with different products. They are targeting lysosomal storage disorder, endometabolic diseases, and other rare conditions, which are approved in all different regions. So, but also very important to us, it's a Chiesi put a big emphasis on patient advocacy, and we believe in listening and really letting patient voice guide our development and access to care. Our vision is all about sustainable solution and working closely with the patients at the global and a local level to improve the life with people for those with the rare diseases. This is included engaging with various organizations, which under the umbrella, this is umbrella organizations such as NORD, CORE, the Rare Disease International, just to name a few, but also disease state-focused organizations such as Jeffrey Model Foundation, Fabry Disease Foundation, Lipodystrophy United, and of course, in the mitochondrial space, International Mito Action, LHON Society, LHON Collective, and of course, we are here on a United Mitochondrial Disease Foundation, like umbrella. So, but when it comes to the topic of this webinar, I want to also provide some update about work that we are doing with the FDA right now to bring idebenone for Leber hereditary optic neuropathy in the US. So I mentioned before, I'm going to repeat again, LHON is an inherited disease characterized by rapidly, progressively, and severe vision loss. And although the disorder can affect both men and women, it usually develop anytime between childhood and old age, and mainly LHON typically affect young adults male. However, women are affected as well, and we want to really highlight that. So idebenone is an oral medication that usually has been taken 3 times and work by supporting the mitochondrial function of the retinal ganglion cells, which also form the optic nerve. So this reduces damage and can improve vision by boosting energy production in these cells. And this giving LHON patients the chance to regain a greater independence and also better quality of life. So idebenone was developed initially by a Swiss company, Santhera Pharmaceutical, and was approved and is been approved in Europe since September 2015. So, and is marketed in Europe and other countries as Raxone. In the 2023, Chiesi gained the worldwide right to Raxone, and we will be working with the FDA since to try to bring to US. So in September 2022, we received from the FDA the news that they accepted our NDA and for oral idebenone for LHON in the US. And this will be based also mainly on the phase 3 RODOS study, which were conducted in Canada, Germany, and the UK. So we enrolled overall 85 patients in this study, but overall, with the package, it's more than 300 patients in other study that we're supporting, that we provide. In the RODOS, though, the age was 14 to 65. They were all carrying at least one of the 3 primary mitochondrial DNA mutation, and with disease onset duration of 5 years or less. So the primary endpoint was best recovery of visual acuity, which was measured from baseline to week 24 in the eye, showing the most improvement or least deterioration. So a higher proportion of patients with idebenone in the group achieved the clinical meaningful improvement in vision versus the patient receiving placebo. So the FDA accept our NDA, and we are expecting the PDUFA date target. It's set for February 28, 2026. And if the application is successful, idebenone will be the first approved therapy for LHON in the US. So hopefully for the next update here, we will provide actually the good news by the meantime. So I'll pass back to you and I appreciate the time. Thank you.
Thanks so much, Giovanni. And yes, it's very exciting that there should be an update in the coming weeks, and hopefully some exciting news to share with the LHON patients that are a part of the mitochondrial disease community. Thank you again. Our next speaker is Robert Fisher. He is the CEO of Omikos Therapeutics, and Robert will provide an update on their clinical trial activities. Robert.
Thank you, Phil, for having me today. I'm Robert Fisher, co-founder and CEO of Omikos Therapeutics, a company in Berlin, Germany. Before I start my update date, please let me wish everybody, on behalf of the entire Omikos team, a Happy New Year, and for, of course, a very peaceful and successful New Year. Yeah, that's just our general disclaimer. Yeah, for those who don't know OMT28 yet, let me please shortly introduce our lead molecule. OMT28 is a small molecule acting as a biased modulator of the sphingosine 1-phosphate receptor 1, and it downstream leads to activation of sirtuin-1 and sirtuin-3, so major deacetylases which regulate function of key mitochondrial proteins. We know from many preclinical studies that this molecule is able to increase NAD+ levels, GSH levels, increases complex 1 activity, improves ATP production, and reduces ROS production, in particular under circumstances of metabolic stress. And of course, we hope and believe that this molecule can help patients to slow progression of the disease, to improve their functional exercise capacity, reduce fatigue, and overall, of course, improve the quality of the life of our patients. So what we did is we first conducted our first phase 2A study and finalized the clinical part just last year. This study was an open label study, which started for everybody with a 3 months natural history part, so without treatment, and then followed by a 6 months treatment part where OMT28 was given once per day as a capsule. We enrolled patients mainly in 9 centers of the European Union. The patients had all mitochondrial tRNA point mutations or single mitochondrial DNA deletions, and from the clinical perspective, they all needed to have, as a clinical feature, skeletal myopathy or cardiomyopathy. And of course, since this was our first phase 2A study, we mainly focused on safety and tolerability, but also wanted to learn much more about many biomarkers and functional clinical endpoints from these patients. Regarding the study population, we saw no major surprises. We were happy to see an equal distribution between females and males. Median age was around 45 as expected. And many of these patients had a cardiac manifestation as it could be expected from the inclusion criteria. And also with regard to the mitochondrial mutations, as expected, we saw major contributions from M3243A2G mutation carriers, but we also had patients with single mitochondrial DNA deletions and other point mutations. Regarding safety and tolerability, this was absolutely confirmed, also in particular in patients with cardiac manifestation. And another focus of our study was to learn more about GDF15, since we thought that and hoped to see that GDF15 might work perhaps as a kind of surrogate marker for clinical efficacy. However, here you see the data for the different time points. What we majorly saw is big variability for these biomarkers. I think other scientists saw these as well already in the non-treatment phase and also in the treatment phase. So unfortunately, no correlation with disease severity scores or motor function tests. So that our conclusion was that at least GDF15 measured in the plasma is not a good surrogate marker for clinical efficacy in this group of patients. However, we were more surprised and positively surprised to see interesting results for main biomarkers for oxidative and reductive stress, so namely NAD+, NADH levels, and GSSG levels measured here in the blood. So you see on the left-hand side an increase in, for instance, an NAD+, NADH ratio in the entire patient population. However, when we look for clinical responders, and we define clinical responders as patients who improved in both motor function tests or the patient global impression of change score, then we saw that this increase in this ratio was mainly driven by the clinical responder group, which we found a very interesting finding and can be overall, of course, interpreted as a reduction of oxidative and reductive stress, in particular in those patients who also had clinical benefit. The same was true for GSH-GSSG ratio. Here, the increase in the treatment phase on the left-hand side is even more impressive in the entire study population. However, again, if we do this split between clinical responders and non-responders, then this increase was mainly and significantly driven by the responder group. We, of course, also looked for motor function tests, and here you see that we were happy to see that motor function improved for the 12-minute walk test and the 5x2 stand test in the entire population over time. Again, if we look for responders versus non-responders, then we saw this significant improvement for the responder group in both motor function tests. The next, what is the outlook? We will conclude this analysis in the next weeks to come, including an in-depth analysis of the responder group. Of course, we want to learn then for our next controlled study, and want to discuss then this next study, the design of the next study, and the endpoints in an end of phase 2 meeting with the regulatory authorities on the US side and the European side. So thank you for all contributors, and back to Phil for the conference.
Thanks very much, Robert. Appreciate the update and look forward to hearing more at upcoming meetings on your progress towards additional clinical trials in this space with this investigational therapy. We're going to move on to our next speaker, Slava Akhmiev. Slava is an Executive Vice President, Chief Operating Officer, and the Chief AI Officer at BPG Bio. Slava is going to provide an update on their efforts to bring an investigational therapy to market for primary CoQ10 deficiency. Slava.
Thank you, Phil, and thank you to the entire UMDF team for having us here today. I will talk about our development program for proprietary compound called BPM 31510, and as it relates to the primary CoQ10 deficiency. Our disclaimer: BPG Bio. Said BPG Bio is a small company located in Waltham, Massachusetts. We've been developing this compound for over a decade, primarily in oncology, and now just recently expanded the use of 31510 in rare diseases. And the first disease that we're targeting is primary CoQ10 deficiency. As you see in the slide, it's a very severe and often deadly disease linked to a number of genetic mutations, primarily in the genes that are involved in CoQ10 synthesis in the human cell. Clinical symptoms here include encephalopathy, ataxia, in general, failure to thrive, and myopathies and other indications, especially kidney phenotype as well. The current standard of care includes primarily oral CoQ10 supplements that have limited success and have been proven to be widely ineffective, especially in severe phenotypes. This disease affects young patients, adolescents, and also adult patients as well. A little bit about CoQ10 replacement here, our compound BPM 31510, that is a proprietary formulation, liquid nanosuspension of CoQ10 molecule. This compound, as I said, has been tested extensively in oncology, and we're actually currently in a phase 2 trial in glioblastoma, where this compound is used as a synthesizer compound for chemotherapy and radiation. That trial is ongoing, and it will be complete by the end of this year in glioblastoma. We started our early access program in CoQ10 deficiency early last year, and it's been actually late of 24. It's been going on for quite some time, and today I will present 4 patient cases from our EAP program and clinical results that we've observed so far. The first patient that's been treated in Germany is in Tübingen, particularly, is a female patient, current age of 9, but she started this treatment when she was younger, about 2 years ago, in 24. As you can see on the right side, we are reporting SARA score dynamic here with this patient, and also FARS ADL, or active daily living activities that this patient reported as well. The first plot here, what you have before the dashed line is historical measurements of the SARA score for this individual. And as you can see, there's a upward or worsening dynamic in the SARA score for this person over the course of 3 years. And then once the patient started on BPM 31510 therapy, you can see rapid improvement in the SARA score, getting towards 3-point improvement by the end of 9 months of therapy. As you understand, improvement of 3 points in SARA score is vastly clinically significant. The same you see in the FARS ADL score. Specifically, I would like to note major improvement in bladder control of 3 points. And if you know that score ranges from 0 to 4. The 3-point improvement is incredibly clinically significant. You see a major improvement in falling, in dressing, and in general handling of daily activities as well. This patient had pathologic variation in CoQ8 enzyme, and I think all of the patients are of the same category of CoQ8 gene mutation. Advancing to the next slide, the patient number 2 is also in Germany in the same institution, also having a CoQ8A genetic mutation. It's a male patient, current age of 17. And we're reporting here very similar metrics of the SARA score and FARS ADL. You can see more modest improvement in the SARA score for this individual, but nevertheless, still an improvement of about 1 point, 1.5 points. That is clinically significant on BPM 31510 therapy. And then what you see on the FARS ADL metric, also an improvement of about 2.5 points, specifically in falling and personal hygiene ability to conduct that independently. Now, next patient here is a patient in the United States in the Children's Hospital of Philadelphia. This is another patient with a CoQ8 gene variation. It's a female patient, a younger patient than previous patient. Currently, she's 11. What you see on the right side, we're reporting MMCOS score progression for that patient. You see historical timeline on the left of the dashed vertical red line before starting on our therapy. And you observe significant worsening, which is the score is going up, worsening of that score with time. And then the orange two lines represent improvement of the MMCOS score over the course of therapy. And then also, which is interesting for the patient as well, the blue line on the right side, if you can see my cursor here, is a bit of regression when the therapy was withdrawn for a person taking a break from personal reasons from that medication, and then going back on therapy again. And as you can observe, an improvement starting again as well. This patient will continue to receive therapy in this year as well. And also our primary investigator who is treating this patient is in the session as well, Dr. Zuela Zilkibi-Cunningham. She might be able to answer any questions that you might have about this particular attempt to treat primary CoQ10 deficiency. The next, actually, very interesting videos of this individual here, this girl before treatment and after treatment. What you can see on the left side, 12 months pre-treatment video that shows very limited ability for this individual to walk down the stairs. When you look post-treatment here in the middle, you can see that she's much more able to walk and much more comfortable walking down the stairs. And then the last video here on the right demonstrates her walking down the stairs independently without holding the hand of the caregiver. And you can see that she's stepping over the step, which is extremely challenging for some of these patients with limited motor skills and the muscle strength. So we're very excited about this result here. As you can see, the videos can tell the story much more quickly than anybody can talk about it. And then the last patient here that I want to talk about, if I can progress. Yeah, this is a patient that's been treated for about 4 months now at University of Bologna in Italy. And the patient is older. It's a female patient, and we have age here. Yeah, 17-year-old female patient. You can see it on the left side. She was wheelchair bound before starting therapy, and then over the course of 3 months, her SARA score had major improvement of 2.5 points, which is incredibly clinically significant again. And now she can stand up and walk after just being 3 months on BPM 31510 therapy. Just to summarize and talk a little bit about our regulatory path, number one is what we've observed so far in these 4 attempts is that this therapy is incredibly safe. We have no serious adverse events reported from any of these patients. All of the patients demonstrate in ataxia and mobility as early as at 4 weeks of being on therapy. We also have reports from parents and teachers and peers that observe substantial improvement in day-to-day activities for all these 4 patients. And to conclude, obviously, that preliminary evidence suggests that BPM 31510 has significant effect in patients with primary CoQ10 deficiency. And currently, we're in active conversations with the FDA. We have a meeting, a Type C meeting scheduled for mid-February to meet with the FDA and discuss our proposal for a phase 2/3 registrational trial that we will begin this year. Thank you, everybody.
Thank you, Slava, and thanks for sharing the additional details on this very promising preliminary clinical data with this investigational therapy. And we do look forward to updates in the future on the clinical trial that's in planning. Okay, we're going to move on to the next organization, Thiogenesis Therapeutics, and we'll hear from Patrice Rioux, who is co-founder, director, and CEO of Thiogenesis Therapeutics. Patrice.
So good afternoon. Actually, it's still morning for me, but I'm in San Diego. So, and Happy New Year to everybody wherever you are. So, I'm gonna do a quick update of where we are at the moment. Thiogenesis, that's a public company, so we have to hold on. Yes, so we have to put all this forward-looking statement. So basically, and I'm gonna go back to some kind of so the rationale, it's going back in time, we know that we are using some form of cysteamine, actually a prodrug of cysteamine. Cysteamine is well known for occurring naturally in humans, and it has been used for the treatment of one disease, cystinosis, and it has been used since 1987 and was approved by FDA in 1994 in kids, in children now starting at the age of 6 months. So it's the I wouldn't say the drug is very safe. As long as you stay in the limit of the design of prescribed actually in the PI, it's a very well-known drug and only expected side effects sometimes. So the way it's used for cystinosis, and that's the key point. The way it's used for cystinosis is that you treat patients based on making sure you don't get side effects, but also making sure you get enough drug, enough concentration to get a biomarker, which is a white blood cell cystine, at a level of less than 1 nanomole half cystine per meg of protein. And that means at the end, each patient has a very different dose. That's a key point. When I was at Raptor, we designed a delayed release formulation of cysteamine, which is Process B. For one reason is that to get this one blood cell cystine actually very low, lower than 1, you need to give the drug. The immediate release has to be given every 6 hours, which is really a problem. And strictly every 6 hours. There are a lot of papers showing that even if you try to go 8 hours, you get the disease progress. So if you want to try that, that's really painful. So what we did with Process B and Raptor, we showed that effectively what is the key point is not to get to a high dose, but to have a continuous exposure for each patient 24/7. And that's why you need this every 6 hours for cystagon or every 12 hours for Process B. On the other hand, this compound, since the 1980s, actually has been tested in multiple indications, and it works almost on everything in animal model, in zebrafish, in rats, in mice, in everything, but never in human. And that's a problem. And that's the reason why I think it's because every time it has been tested in human, it has been tested, including when I was at Raptor and we tried that in mitochondrial disease or NASH or anything like that. We actually try to develop the drug based on the standard way of doing it. That is, try a low dose, medium dose, and high dose for everybody in a group, which basically, if you come back to what I said earlier, we were not giving each patient a specific dose. So to get there, the idea that instead of continuing to try to develop these drugs that works for any animal but never in human, we have to go back to, can we use the same mechanism of action for all other indications? And if so, is there a way first to get the drug 24/7 without all this problem of every 6 hours or every 12 hours and a peak? The reason for giving every 6 hours or every 12 hours is that because the drug is eliminated, cysteamine is eliminated very quickly, so you have to use a very high dose to make sure you can cover. So is there a way of getting a continuous exposure without the peak? And then if it's possible, can we determine a biomarker that could allow us to determine the dose? So just to remind very quickly remind you exactly in cysteamine, in cystinosis, what you have on the top right is the way it works for cystinosis, and it has been like that for more than, as I said, since 1994. So the way that cysteamine goes into the lysosome breaks the cysteine into cysteine and then allows the cysteine to get out through the lysin transporter or through the cysteine, because in cystinosis, the transporter for cysteine is broken. But what if it actually works the same way in patients who are not cystinosis patients? And basically, we think that it's the way, because when you give cysteamine in certain amount, you increase the level of cysteine, which is the thiol versus the disulfide. Cysteine is a key parameter to build glutathione. When you have glutathione is the best antioxidant for treating ROS in mitochondria. So I don't want to go into many details here. So what we did first is that we designed a new compound, which is a prodrug, and which is two thiols, but instead of a homodisulfide, it's a mixed disulfide. And what you give this thiol, which is TTI-0102, this disulfide, you give it orally. It's never absorbed, which is very interesting. It's never absorbed, but it's actually broken first by reduction into one cysteamine and one pantothecane, and then the pantothecane itself, it's also not absorbed, and it's broken again in cysteamine and vitamin B5. So basically what we get from that is that we get, as you can see here, a nice green curve for 2400 meg versus what we would get for 600 meg of the immediate release, with the same peak, despite 4 times the dose, and a continuous exposure for 24/7. So based on that, we initiated a study in Milas in Europe. So Milas, I won't go into details. Everybody knows actually in the group, but we submitted an IMPD in Europe with TTI-0102 to treat Milas, having in mind that we know very well the safety, and so we could go relatively quickly even in children. At the moment, we have been treating only adults, but we treated actually 9 patients. Importantly, it's the fact that some of these patients were relatively small, 39 kilos, and some were at 82 kilos. And we had 6 treated with TTI-0102, 3 with placebo. They were treated first with 1 week of half the dose, and then everybody had the same high dose, 5.5 grams, because we wanted to look at the impact of one relatively high dose on biomarkers in order to find a biomarker that would correlate potentially with, or could be predictive with, a clinical endpoint, and would allow us to determine a dose systematically per patient, as we could get from the cystinosis. So we measured PK, PD, tryptophan, pantothenic acid, GDF, FGF, all this stuff. Also clinical efficacy, and we'll have the results presented actually in 2 weeks. Yeah, 2 weeks in PISA. And at the same time, we submitted an IND for Lee syndrome, which is kind of similar mitochondrial disease with also the same problem of ROS, of oxidative stress. And these patients, this study we have the IND, so it's going to be 2-stage phase, first phase 2A, where we start with double blind with adults and adolescents, and then a second part with children. So, and even based on the patients on the first study we're going to present, actually, we think we could even go probably faster in the stage 1. So that's something to discuss later on at the next meeting, probably. Having in mind that the goal would be to have kind of phase 2b/3 where we could have kind of even basket study where we could have MILAS and Lee syndrome at the same time in the US and Europe if we can do that in parallel. And that's it. Thank you. Thank you very much. And if you have time to go to Pisa.
Very good, Patrice. Thanks very much for the update. Congratulations on the progress there, and we do look forward to hearing continued updates on progressing
Yeah.
this for both MELAS as well as Lee syndrome spectrum patients. Okay, we're going to keep moving along here. Our next presentation is from Ash Dugar. He's the Chief Development Officer at Pretzel Therapeutics. So, Patrice, we'll need you to stop sharing.
Yep.
Thank you.
All right, excellent. So, hi everyone. Happy New Year. As Phil said, my name's Ash Dugar. I'm the Chief Development Officer at Pretzel Therapeutics, and I'm joined by Julie Jones, our program team leader and VP of Biology, and actually someone who's been working towards treatments for mitochondrial disorders for years, and I believe this community knows her very well. I just want to thank Phil, Cara, and the entire UMDF team for inviting us to speak today. While I've been doing drug development for about 25 years, and most of it in rare diseases, I'm pretty new to rare genetically driven mitochondrial disorders, and so I feel incredibly privileged to talk to all of you and have a chance to learn from you. These are forward-looking statements. If you're interested in more details, you can visit our website. For those who have not heard of Pretzel Therapeutics, we're a small biotech company based in the US and Sweden. Our mission is to create a new paradigm in the bioenergetic space through transformative mitochondrial DNA approaches to treat patients with areas of high unmet medical need that have mitochondrial DNA depletion syndromes being a priority, starting with POLG disease. And this is what drives us at Pretzel each and every day. So our flagship program and what we'll focus on today is PX578, our lead clinical stage program. It's a small molecule, brain-penetrating POLG activator, and we are currently in phase 1 in healthy volunteers. Now, we're incredibly excited about this program, and I'm going to show you just a little bit of the non-clinical data that we've generated in patient cells and in animals. We have substantial preclinical efficacy in multiple in vitro, meaning in cells, and in vivo models of MDDS or mitochondrial DNA depletion syndromes. We've seen that in non-clinical disease models, PX578 can increase mitochondrial DNA when it's depleted due to various genetic mutations, and thereby increase oxidative phosphorylation. We've shown efficacy in 2 different MDDS mouse models as well. And very importantly, especially for those individuals with POLG disease, is that this compound is CNS penetrant. It does cross the blood-brain barrier. Of course, we're focused on not only the brain, but the rest of the body as well, as nearly all systems in the body are affected. Over the next couple of slides, I'll show how PX578 is working in both cells and animals. The graph on the left shows a biochemical assay evaluating the effects of PX578 on 3 of the most common POLG mutations, and you can see the response to PX578 in terms of increased POLG activation. Currently, we've tested PX578 in over 50 mutations, and it has shown a response in the form of biochemical potency to all of them. This is the first step, if you will, demonstrating that we can meaningfully activate POLG. In addition to biochemical potency, we look at patient-derived cells in the middle chart. Here, we bring cells from several different patients carrying POLG mutations into the lab and look at the response to PX578 in vitro in those cells. We experimentally deplete the mitochondrial DNA from the patient fibroblasts and evaluate its mtDNA recovery with or without PX578. The cells depicted in purple are treated with PX578, and as you can see, we have robust restoration of mitochondrial DNA copy number. In addition to measuring mtDNA, we explore how the cell is producing energy to see if we can improve cellular respiration. So on the right side, in the rightmost graph, we measure oxygen consumption rate, or OCR. The increase in mtDNA observed in the middle graph correlates to an increase in OCR depicted on the right-hand side of this graph, on the right-hand side of the slide in the graph. And we went further to exemplify the power of our approach in much more energetically demanding cells, such as neural stem cells carrying POLG mutations. Again, we see a robust restoration of OCR upon incubation with PX578. So we really have the ability, and we've shown positive and robust data in multiple non-clinical models. And on the next slide, please. We have some very exciting data from one of our animal models. Here I'm going to show you some data on our TK2D mouse, a model of MDDS with severe mitochondrial DNA depletion, which leads to a decrease in survival. Now, TK2D mice die within a few weeks of life, shown by the black line. In our study, we started treating these mice on day 4, and as you can see, we get a significant increase in survival of these mice as shown by the blue line. This model has been used in evaluating nucleoside therapy for TK2D patients, for which UCB gained regulatory approval in the US recently, and this is incredibly exciting for the community. On the right-hand graph, we see a significant increase in copy number of mitochondrial DNA using samples taken from the liver. We have additional data in cells and in other mouse models, such as the DGWOC knockout, where we show an increase in mtDNA and resultant functional benefit. I do want to just take a second and say, with the support of the POLG Foundation, we're in the process of evaluating a POLG specific mouse model as well. And now what's really exciting for PX578 is that we're in the clinic, and I'll provide an overview of that progress. So in early 2025, we started our double-blind randomized phase 1 study in healthy volunteers, and that study is ongoing. We look to complete phase 1 in the first half of this year. Now, following the phase 1 study in healthy volunteers, our preparations are on track to start our first study in patients with PX578 and POLG disease in the second half of 2026. And our goal is to generate some early safety and clinical data by mid to end of 2027. We are laser focused on initiating our global registrational study this year to support global regulatory approvals and ensure a best-in-class profile with meaningful improvements in clinical and patient-reported outcomes supported by biomarkers. And as we get closer to that first in-patient study, we will share more details with the community. And with that, I'd like to thank you for taking the time to hear about our progress. And again, thank you to UMDF for hosting us.
Well, thanks very, very much, Ash, for that update. And we're really pleased to partner with both Pretzel Therapeutics and the POLG Foundation to advance natural history study data collection here in the coming years that we hope will be so supportive of the development of therapies for POLG disease. So thank you for your partnership. All right, we're going to wrap up today with our final presentation from Dan DiPietro, who is co-founder of Pierpont Therapeutics. Dan, I'll turn it over to you.
Thanks, Phil. Good to see everybody, and good to hear the presentations. And I also apologize because I'm getting over a cold as well, so bear with me. So Pierpont Therapeutics, this is an opportunity for us to kind of introduce the company to the community. It's a relatively new company, but the principals who founded Pierpont Therapeutics are biotech entrepreneurs who've been in the mitochondrial disease space for some time. The principals at Pierpont Therapeutics founded Modus and led Modus Therapeutics, which developed the nucleoside therapy that was just approved and just mentioned by Ash, ultimately acquired by UCB, and the approval was just announced in November. So it was an extremely gratifying experience working on that program. We are continuing this focus on diseases within mitochondrial disease that have clear genetics and pathophysiology. We believe that this increases the likelihood of success by having a better stratified patient population to test products in that have a better biological rationale. We were also very gratified to receive the inaugural investment from the UMDF Venture Philanthropy Initiative, and continue to work closely with the UMDF on a number of programs. We believe this product that I'm going to describe has a clear path to approval. It has a very unique biomarker that's disease specific. Unlike most in mitochondrial disease and in other diseases, this is a very disease-specific biomarker that we're using for both dose dose identification, as well as utilizing it for regulatory purposes as a surrogate endpoint. We are IND ready. We've completed IND enabling studies and are gearing up for clinical trials, hopefully by the end of this year. So the disease that we are developing our product, which is called PTI 501, is called Mingi. Mingi is a monogenic disease caused by autosomal recessive or compound heterozygote mutations in the thymidine phosphorylase gene, which encodes the enzyme thymidine phosphorylase. Thymidine phosphorylase is responsible for catabolizing excess thymidine. And when patients have mutations in this gene, they have excess thymidine, which causes a DNTP pool imbalance, similar to TK2 in a lot of ways. Except in this case, the ratio of pyrimidines to purines is skewed toward the pyrimidines. When you have a pool imbalance in the nucleosides in the mitochondria, the mitochondrial DNA replication is impacted, and so patients show in affected tissues mitochondrial DNA depletion, deletions, and point mutations. And the unique aspect of Mingi is that excess thymidine is present in the blood and in the urine, and so it offers a very disease-specific biomarker that one needs to reduce in order to have an impact on the disease. The thymidine is elevated in Mingi patients universally. It's in excess of 3 micromolar in the blood and in the urine, whereas non-Mingi patients have no detectable thymidine. There is a blood test that's commercially available, which we're going to be utilizing for patient identification. We believe Mingi is highly underdiagnosed, which we'll talk about in a second. But the blood test, which is just an analyte test, enables, we think, broader outreach to detect undiagnosed patients. Genetic testing can also be used, but having the blood test and urine test, we think, is a very interesting feature of this disease. And like I said before, it represents the ideal biomarker and a surrogate endpoint for clinical development. Just very quickly, Mingi manifests primarily as a gastrointestinal disease. Patients have malabsorption. They're very underweight. They lose muscle mass, and patients also develop, secondary to that, those GI symptoms also develop peripheral neuropathy, hypotonia, and often are wheelchair bound. Diagnosis is typically in the early teens, but we think the patients are starting to show symptoms much earlier than when they're actually diagnosed, as most of these patients are going through a diagnostic odyssey, very common in the rare disease world. And the disease is fatal. Patients typically die in their 20s or 30s, and usually the cause of death is secondary to the GI effects, the malnutrition, bowel perforations leading to infections, etc. And there are no pharmaceutical treatments available, so this is, by definition, a huge unmet need. So our product is an enzyme replacement therapy. We are replacing the thymidine phosphorylase enzyme to patients who have a mutated form of that enzyme. Similar to some of the lysosomal storage diseases, which has really transformed that field. So it's a tried and true method of treating diseases that have monogenic causes. The enzyme itself is the thymidine phosphorylase human enzyme, and it's conjugated to a cell-penetrating peptide, a proprietary cell-penetrating peptide, which enables broad tissue distribution entry into cells and then the release of the payload from the endosome. It enters the cell via endocytosis, but then the CPP12, which is conjugated to our enzyme, releases the payload into the cytoplasm, which is where thymidine phosphorylase is active natively and where the catabolism of excess thymidine needs to happen in Mingi patients. The removal of the systemic thymidine. So again, these patients have elevated thymidine in plasma, but also in tissues. There have been other modalities used to reduce thymidine transiently in the periphery and have shown a great clinical impact. And so we're attempting to do that with thymidine with PTI 501. So it's getting broad tissue distribution as well. So it's getting into the tissues of interest as well as into the periphery to reduce that excess thymidine. So again, the Mingi is unique in that it has this causative biomarker that is elevated in plasma and tissues. So it's across the board universal, and Mingi patients have elevated thymidine. So the goal of therapy is to reduce thymidine both in the periphery and the tissues of interest. We've tested the enzyme in a mouse model of Mingi, which show elevated thymidine, and we've shown a potent reduction in peripheral and in tissue thymidine levels, which we'll show in a minute. And this mimics some of the clinical interventions that have been tried in Mingi, which include continuous ambulatory peritoneal dialysis to physically remove thymidine, bone marrow transplants, and liver transplants, which are also effectively delivering wild type thymidine phosphorylase to remove excess thymidine. And those modalities have all shown significant clinical benefit, but they all come with some issues, particularly bone marrow transplant and liver transplants, because these patients are very fragile and aren't necessarily candidates for such a traumatic intervention. Where CAPD, which is a lot more friendly on patients, has shown significant clinical benefit when removing thymidine physically. The biomarker data and the clinical improvement that we've seen in using these other modalities makes us very confident that utilizing an enzyme replacement therapy to try to achieve the same thing, but in a much more patient-friendly manner. This is going to be a subcutaneous injection. Has a great chance of success. The biomarker itself, given that it's disease specific and is causative for the disease, we believe is very consistent with the biomarkers that the FDA has been recently talking about as good surrogate markers for accelerating the development of rare disease drugs, particularly the newly announced RDEP program, which really talks specifically about disease-specific biomarkers and the ability to more quickly approve programs that have been shown to modulate disease-specific biomarkers. One of the things we like to talk about also is the mechanism of action. So we're reducing excess thymidine to rebalance nucleotide pools. So, like I said before, the mechanism of Mingi is you have excess thymidine, which causes this nucleotide pool imbalance in the mitochondria, where the ratio of pyrimidines to purines is too high. And so our goal is to rebalance that ratio so that it's closer to the sort of 50-50, which is in the healthy state, which is the case, which is the ratio in the healthy state in mitochondrial. The DNTP pools in mitochondria are typically 50-50. And so we mentioned, the previous speaker mentioned the approval of Kygevvi for TK2 deficiency by UCB, which is the program that the founders of Pierpont started, is attempting to do the same thing. And we think it validates this approach to when you are able to rebalance nucleotide pools, you can have a massive clinical impact. And so in the case of TK2 deficiency, the patients have low amounts of thymidine monophosphate and cytidine monophosphate. And so by providing the substrates of DT and DC, you're able to restore that pool imbalance. And you can see the survival advantage that this is from the label, the prescription label for Kygevvi. You have a massive improvement in survival. Really, the survival curve is really just astounding. And really, we believe that this rebalancing of nucleotide pools in diseases of mitochondrial depletion disorder that are due to mutations in enzymes responsible for nucleotide metabolism have a great potential for providing clinical benefit. And this is the preclinical data that I mentioned before. This is the Mingy mouse, where you've had the TIMP gene mutated, so the mice have extremely high levels of thymidine. These mice have been delivered or have been administered our enzyme, and you can see the gray lines are the pre-treatment, and the blue dots are the thymidine levels after treatment. And you can see that we are able to reduce excess thymidine very robustly in this mouse model of the disease. And importantly, and this is in the blood. Uh, Importantly, we also think we need to reduce the levels of thymidine in tissue, not just in blood. And so we also tested whether or not our enzyme was able to reduce excess thymidine in tissues, particularly the tissues of interest in that are affected in Mingy patients. So this includes the small intestine, the liver, and as well as the spleen, heart, and brain. And you can see across the board that the levels of thymidine are reduced significantly and robustly. And this is just 1 day post PTI 501 administration in these mice. So we're having a great biological response in this mouse model of the disease of interest. One of the other things I wanted to just let the community know is that there is a academic sponsored or academic run retrospective natural history study that is just getting started. We did our own analysis of the published literature and found that although if you do a PubMed search for Mingy, many of the publications mention that there are about 200 patients identified in the literature. When we did a deep dive into the literature, there are actually greater than 700 cases in the published literature, and they have a lot of good information on the disease course. And so this natural history effort will increase our knowledge of the disease progression. This will be of use for any products being developed for Mingy, and also facilitates the patient identification for current and future trial recruitment. And we're also interested in creating a patient registry, ultimately, who will be the patients who would be candidates for therapy. And we also believe, given that this disease we think is extremely underdiagnosed, the outreach to Mingy physicians and researchers will help with understanding just where those patients are and how they're being treated. This was our initial clinical trial. We're anticipating that this product, the target product profile, given the preclinical data, is that this is a once or twice weekly subcutaneous administration. So we believe that this would be administered by the patient or by a caregiver. No visits to the infusion center are necessary. And we're going to be dose titrating each patient based on that plasma thymidine biomarker, which is an extremely powerful method to ensure that each patient is receiving, the right dose. We want to demonstrate a clinical benefit in the GI and neuro symptoms, evaluate the long-term response in the clinic as well. And we're anticipating that this will include 10 to 15 Mingy patients. And the main goal is really to see that biomarker response and see a sustained reduction in that plasma. And our secondary goals in this study will be to look at clinical endpoints as well. And we're hoping that this trial can start sometime toward the end of 2026, but we'll keep people posted as we roll that out. And just want to conclude by saying we're happy to be working within mitochondrial disease again. The Modus experience was extremely gratifying one. To see that product finally get approved in November was, extremely exciting. And our product right now, we believe, has a very high likelihood of success, similar to that one, in that we have data in a robust animal model. It's an ERT, which has a track record of success in other disease states. And we think with the biomarker, we're going to have an extremely efficient and abbreviated clinical development program. And again, that retrospective natural history study is just getting up and running. And so for many people on the call, academics, you'll start to receive emails asking about patients. So thanks again for the opportunity to talk about Pierpont.
Yeah, thanks, Dan. And thanks for all the efforts that you, your collaborators, your partners have put into not just Mingi, but of course TK2 deficiency as well. We're really excited about the prospects for this approach to treat this huge unmet medical need for Mingi patients and the terrible disease that results from it. We're going to wrap up for today. I want to thank all of the speakers from all the organizations who were on today, but also last month. Over these two webinars, we've heard from two companies with approved therapies to treat mitochondrial disease patients. Today, we heard from two companies that hopefully will have approvals from the FDA to treat mitochondrial disease patients. And we've heard from a dozen other companies that are actively developing therapies to treat mitochondrial disease patients. It's an incredibly exciting time. We're still in the early days of the clinical era for mitochondrial medicine, but it's a really hopeful time. So for the patients on here, thank you for those who have participated, whether in MitoShare as a registry or in a clinical trial. We're going to need your engagement to continue to be successful here. But please know that UMDF mission is to assure that there are effective therapies for all forms of mitochondrial disease and for all mitochondrial disease patients. And we're on the path to making that happen. So thank you again for your participation. A quick reminder that next month we'll be back for our Bench to Bedside webinar. We're actually going to hear from Michelangelo Mancuso, a summary of the Mitocon conference that Patrice referenced earlier in this call, so that we can hear the latest from what came out of that European meeting. And that'll be on Monday, February 9th at 12 p.m. Eastern. Again, thank you for all who are here today, as well as maybe watching this, the recording, and we look forward to seeing you at a future Bench to Bedside webinar. Thanks, everyone. Happy New Year.